as if you're seeing my screen. At least it shows so. So I am Jas. My name is Jas, and I am professor of environmental engineering in Denmark at a small, obscure university somewhere in the northern part of our small country. And what we have been focusing on in my lab is basically to analyze microplastics in complex matrices in the environment. So we started out and still use quite a lot the micro FTIR imaging approach for that, but we are not only using that because we are focused on basically the system because environmental engineering and so on, right? So what we want to do is to get from a complex matrix, like for example this harbor sediment you see over here, or whatever the fishes or the sludge or the soils, to some numbers. And I'll come back to that. But what we want is we want partly to know how many pieces of plastics there are in a sample. We also want to know which size they have, which shape they have, the morphology, right? But we also want to know the mass, and that is a bit tricky. But I'll come back to that. So we want to go from some really nasty stuff to some nice numbers. And to do so, the first thing you have to do is to get the sample collected. And for that purpose, we have been putting quite a lot of energy in building various types of sampling equipment. For example, this system, we call it the UFO system. We like nice names. Universal Filtration Object is what it stands for. With this piece of equipment, depending on how we have constructed it, you can filter 1 cubic meter in 1 to 2 hours on a 10 micrometer filter. You can also put a smaller filter in, a 5 micrometer, but that would of course clock faster. So this is a portable unit, and you can carry it if you have strong arms. It's pretty heavy, and then you can go out and filter. But what if you are somewhere else? What if you are in the marine environment and you want to profile the deep sea? If you are on a big ship and you want to do sampling of the water, you have to and you want to do it in 2 small sizes, you would use in situ pumps. And the problem with the in situ pumps you actually can buy, there are some you can buy, is that they are filtering quite slowly. When you have a ship which costs you maybe, I don't know, $50,000 a day, lying out there, you cannot lie on the same location for hours and hours in order to take a sample. You want to be fast. So we built some very fast sampling equipment where we can filter like 3 cubic meters in half an hour. This equipment you see here can go down to 200 meters. We're building one now that can go down to a kilometer. So we're also sampling other places, places which are easily accessible in many ways. For example, soil samples. What you see here are the badlands of Iceland, the deserts of Iceland, where we're sampling some weeks ago, for example. So you have there. It's pretty easy. You just have to use a shovel. But you have to get there, of course. We're also using filtering for analyzing, for example, drinking water. Drinking water is a comparatively easy matrix. There's not much in it, but you have to be extremely careful with respect to contamination and so on. So we have also tailored devices for sampling there. So it starts with the sampling and sampling the right amounts of matrix and doing it without contamination. What is important when you're sampling is that you sample enough atoms of plastics. It's not so much a question about how much volume you end up sampling, but that you sample enough material, right? And often you don't know exactly what enough is, so you have to sample a lot. For example, drinking water, we also typically take a cubic meter, maybe 2 cubic meters, and filter them on 3 micrometers or 1 micrometers metal filters. Once we have collected our samples, the work has just begun, because then we have the whole matrix, and I mean, fortunately, there's a lot more out there than plastics, right? So you have to get these very often very low concentrations of plastics, like ppm or ppb or whatever, out of the matrix. and that can be quite challenging. So this is an example of getting a sample. For example, this is a marine sediment sample where you have collected with a grab from a ship, and then we have to analyze that, to degrade it, and then we end up at some point of time with our plastic extracts in these small vials. I'll not go into too much detail about it, just a little bit about how we do. But this step to get from this sediment to the concentrate is really the big work. I mean, for a sediment like this, it takes 3 months to prep it without damaging the plastics. So this is really the big thing. And then we analyze it. We analyze it with different machines. The machines we mainly are using are the FTIR machines, but we're also using pyrolysis, GCMS, and we're also using Raman. But the workhorse and the one I will talk about here is the FTIR. I think the message is that different machines can do different things, so it's not like, should I have this machine or that machine, or what is the better machine? It is a question about what do you actually want to get out of it, what is the purpose of it, and what is then the suitable approach for that purpose. Just to get back to the sample treatment, we are following standards, basically, or not standards, because there are no standards in the field, but we are following what everybody does. We are often doing pre-oxidation first, and then we are using a number of enzymes depending on the material we have to digest, and then we are using oxidative treatments to remove the materials. And this is an example of the oxidative treatments. This is a Fenton. And for those of you who have tried to do Fenton oxidation, you know that you have to be a bit careful because it gets pretty hot, if I may say so. So if you're not careful, it'll explode up in your head. But once you mastered how to do it, this is a pretty efficient and also pretty gentle way to prep your sample as part of prepping your sample. But you also have a lot of inorganics in your material, and you have to get rid of sand and silt and clay. And I mean, getting rid of sand is a piece of cake, but getting rid of the clay and so on and the silt can be very, very demanding. So we have different density separation steps. Depending on what we are analyzing and how we are analyzing it, we apply them in different parts of the protocols. But we're using different chemicals for that. We are mainly using zinc chloride or a sodium polytungstate, depending on what we're doing. We are not much using sodium iodide, but I mean, it's not that we haven't used it, but we're mainly using the zinc chloride and the sodium polytungstate. They have pluses and minuses, so again, it depends on the job, what you want to do. This is an example of such sedimentation. On the left, you see the post sedimentation, the post fractionation we are doing, where we want to get rid of the last bits of clay and so on in the samples. So we leave them there for a day or 2 days, and then they'll settle, and then collect the floating materials, and then we analyze them. If we have really big samples, we are using something we call the rocket, which can take quite a kilogram of material and so on. But again, things have pluses and minuses, and sometimes it's easier to use the small funnels and have more of them than using the big machine because it's heavy to operate. So, for example, when we process a kilogram of sediments, we will first pre-oxidize it, and you have to pre-oxidize your sediments because your plastics will be baked into a matrix of clay, organic matter, and so on. And you have to break that up in order to get your plastics out, or some of them will be baked into that. So you have to do some pre-treatment, and we do that with peroxide. and then we do a flotation to get rid of most of the stuff, of the heavy stuff, of the sand, and so on. And then we do a number of these enzymatic and chemical approaches, another flotation, and then we collect the samples in our small vials, right? So after having done all this, which is really the like 80-90% of the work, we have particles in two size groups, and when we are going for the FTIR, we sort anything above 300 to 500 micrometers manually out of the samples. Why? Because they are too big for the micro FTIR imaging. I'll come back to that. And these big particles we then analyze under an ATR, under a simple FTIR ATR. Just take one piece and put it under and get the spectrum, and then compare to a library. So that's pretty straightforward. It's pretty tedious. It takes a long time if you have many particles to weight them, measure them, and get the spectrum and so on. But this is what we do with the big particles. With the smaller particles, we instead are using an imaging approach, and we have our sample on a window, and that window we then scan. And we are using a focal plane array, but you could also use a linear array for the same purpose. The key here is that you get a map of spectra, like in the picture you see here below. So you get a map of spectra for each pixel on your image. You have an FTIR spectrum, and then you have to interpret this. And what we have is two machines with 128 FPAs, so that's the biggest you can get. And we can go down to spatial resolutions on the pixel of around 3.3 micrometers. This is not the same as the size detection limit, but this is the pixel resolution. So for the small particles, again, we take our concentrates in the vials, and then we deposit them on the window. Some people use filters, some people use windows. We use zinc selenide windows mostly, not only, but mostly, because they have a good, they don't absorb IR in the range we are looking at. And then we scan them with our machines, and a scan like this takes 4 to 5 hours. And out comes, in this case, if we scan it in our normal operation, 3.2 million spectra making up the map. Each spectrum then looks something like that, whatever, right? And then you have to interpret that. Oh, by the way, again, just a very short note on what you sort of scan on. It depends on your purpose. And we are using different types of materials to scan on. So again, it's not cast in concrete. It is something which is depending on what it is you actually want to do. After having done a scan, as I said, we have the 3.2 million of spectra. And in the beginning, what we did was we took each and every particle and then looked at it. So we had this map. This on the left you see the visual image. On the right you see an FTIR image, basically a false color image. And then we clicked on each of these many particles. If we had to go through the whole window like this, it took about 1 month to analyze one scan. And that started to annoy people quite a lot, or better, we were not able to do it. So we had to do something about it. And what then came was some good colleagues and friends of ours in Germany, Sebastian Premke and company. They made an automated algorithm, and we saw, oh, that was smart. And we built a similar algorithm and called it Simple, which today is joined with the work from Sebastian Premke at the Wegener Institute in a common approach, which is meant to be MP analysis for non-nerds, meaning that it helps you analyzing these millions of spectra. And it is a freeware. Anybody can download it, and there's also databases, free published databases available, which you need for this purpose. In the software, we do different things. I will not go into too many details here, but for example, we calculate the longest distance, the length of the particle. If this is in the particle you see here, for example, this one, right? We calculate the length, and then we want to find them. We basically want to go from the 2D to a 3D estimate. So we want to see if we have this particle, how big, how much volume would it have, how much mass would it have. And of course, we only have 2 dimensions, so we have to estimate the last dimension. We do that by simply assuming that this is an ellipse. So we calculate the equivalent ellipse of this, and then we assume that the minor dimension of the ellipsoid is a certain fraction of the minor dimension. So we estimate from this image this ellipse, which is of course an estimate on an estimate, but still it gives an idea about what the volume is. And based on that, we then have the polymer type and we calculate the mass, because I think it's super important to have the mass. So we need both the particle numbers and we need the particle mass. This, by the way, works really well for some particles and not so well for other particles. For example, a fragment like the one you see on the left works fine. A fiber like this doesn't really work super fine. So it's not a silver bullet, right? But it gives a reasonable estimate. So why do you want both size and mass? I mean, nearly all the published studies are on a number of microplastics in a certain size range. For example, I found 500 microplastics between 50 and 500 micrometers. But this is really a tricky information. Because think about it. If you take, for example, a particle of 1 millimeter times 1 millimeter times 1 millimeter, and you take your magic hammer and give it one on the head with your magic hammer, which creates 10 micrometer particles, what you will get out is 1 million particles. If you make one micrometer particles, you'll get a billion out of it, right? So you have to remember that there is no law of conservation of particle number. There is, on the other hand, a law of conservation for masses, at least to some degree, assuming it's not degrading. So in order to make a lot of different types of estimates, for example, which is the bigger source, you must have the mass. You cannot do it on numbers alone. On the other hand, numbers, mass alone is not sufficient neither, because size matters. Think about the Daphnia we saw in the last presentation. Assume there come a rubber boot, the Daphnia cannot take it up, right? So size matters. Size matters. So you need both informations. This is just to show a little bit of some study we did. An example of microplastics in indoor air. This is just to show some of our work, where we had a breathing mannequin. So this is a breathing mannequin in a room, and you see the mannequin's lungs here, and the mannequin is sitting there, and then there is a filter in the lungs of the mannequin here. It's a silver filter put in here. And then you can see a scan of the silver filter afterwards here. And we had this in 3 different flats for some time, and we measured what is the plastic content in the air that you and I and everybody is breathing. And this is a scan of something like that. And what you can see, the green stuff is plastics. It's mainly polyester. And the gray stuff is different forms of different forms of organic materials, right? So this is for example a cotton fiber, and what is this grayish here? Yeah, the grayish stuff is proteins, and you know what? They come from your own skin and your own hair. So about 90% of the stuff you breathe in, the dust you breathe in, is your own dead skin. Some is cotton, and maybe a handful of percent is microplastics. Okay, so you breathe around 5% microplastics in the dust you're breathing. We are only looking at the organic part of the dust here, by the way. And this is, of course, varying between the different flats, right? But the general picture is like that. Very short before I close, I just want to mention something, and that is a discussion about size detection limit, which is I think it's important to take when you're discussing about plastics. You have to remember that when you are using especially the spectroscopic methods, it's not so problematic for the pyrolysis, the thermal desorption methods, but for the spectroscopic methods, it's really important that the fact that you have identified one particle of some size does not mean you have found them all. Right? If you find a small particle of, I don't know, 50 micrometers, it doesn't mean you found all 50 micrometer particles. So this is not your size quantification limit. And when you are going to the manual sorting approach, like I showed we did for the bigger particles, it's quite difficult actually to state what is the size detection limit. But it's very often overestimated. People think they can analyze down to lower numbers than they actually can. And people are not really, commonly also not reflecting enough about this issue. I mean, it's totally fine to use manual sorting and analyzing, but you just have to be aware of the limits of it. I would just want to show this as an example, and maybe this is just on the closing slide, and I will jump over the rest of my slides because I'm talking too much. This is a study from China, which actually was very nice. It was very well conducted and very thorough. It looked at the sludge from 28 Chinese wastewater treatment plants, and they determined the plastics in the samples by sorting and manually analyzing. This gives you in this case an estimate that you had about 1.5 times 10 to the 14th pieces of plastics per year coming from all of China's wastewater treatment sludge. We did a study we just published on one Swedish treatment plant. I mean, it is a big one. It is the second largest in Sweden, but still, and we found the same number of plastics. So, does this mean that one Swedish treatment plant has as much microplastics as all Chinese treatment plants put together? No, of course it doesn't. Of course it doesn't. It means that the two different studies have two different size quantification limits. So this is really the danger when reporting in numbers, because there are many more of the small particles than there are of the big particles. So it's not as problematic if you have mass. Then you would have been much closer in the comparison. Also, if you had only looked at the big particles. So I think it's super important to remember both mass and size matters. So I'll just jump the last slides now because otherwise I will never get finished. So that was my and thank you for being allowed to do this talk. And yeah, questions, please.
Thank you, Professor Vollertsen. Am I audible?
Yeah, here you go.
Yeah, sorry I couldn't introduce you. Thanks for your nice presentation. It was really interesting, especially the spectroscopic and image-based analysis of the microplastic samples using advanced IR imaging techniques. There are a few questions in the chat box which I can read over to you. Could you please explain what safety measures you have adopted while performing phenton oxidation? Does it explode even at this low temperature of 15-30 degrees that you have mentioned?
I mean, we always do it in the fume hood, and we have it on ice. So when we are doing the phenton, we have relatively small volumes, because if the volume is too big, you cannot cool it. And then we have it in an ice bath, and we have it in a fume hood. I mean, today it doesn't explode for us any longer because we know exactly how to do it. But in the beginning, it was actually, it happened several times before we learned it the hard way. So have it in a fume hood and keep it on ice. And then we just are standing and looking at it for the first couple of hours until it settles down, and then you can leave it for the next number of hours, 5-6 hours or a day or something. There are also some other issues with it. You have to, if you're not careful, you will get the precipitates. So this is also something you have to fine tune. If you just leave it too long, you'll get precipitates.
And what is the source of your drinking water? Is it commercial drinking water?
The sampling you saw up there was from Sweden. And this is from surface water reservoirs. In Denmark, we also measured on drinking water, and we're using groundwater.
Okay. Using which method you have separated inorganics from the filter paper?
We are using density separations, meaning the first thing we do is we do pre-oxidation to break up the matrix. And then if it's a soil or sediment, and then we do density separation. This pre-oxidation takes any time between 1 day and 1 week, where we continue to put peroxide on it until it stops bubbling, and then we floated it either in zinc chloride or sodium polytungstate, typically on densities around 1.7.
What is the average analysis time if you were to compare the particle sizes, let's say 100 micron particles versus a 10 micron particle? What would be a typical analysis time?
I think it cuts between the manual stuff you have to do manual and stuff you can do on the scan, and one scan takes about 5 hours. We typically do. We often do several scans, maybe 3 scans, but at the end of the day, it's a question about how many particles we find. On the other hand, if you have the big particles, if you, for example, have a manta trawl from the marine environment and have to go through all the particles there, it's really taking a long time. I think the key is that it's a sample prep that typically takes the longest time.
Okay.
But in many ways, it's easier to do the small particles than the big particles.
Okay, I'm seeing a lot of questions coming up. Maybe we'll have last 2 questions before we close this session. What kind of microplastics occupy the largest number in wastewater sludge? I'm sure this is.
It differs. Yeah, I mean, there's a handful or so of the plastics you always find, and it differs. I mean,
Okay.
It's really difficult to say.
I have one last question. So the FTIR-based imaging technique that you demonstrated was really interesting, wherein you really scan something around 1 million IR. I mean, you take infrared, spectra over the complete sample, and then you try to understand the polymers, right? So, is it like a software that is available, with the standard library?
Yeah,
that can be.
Yeah. It's freeware, and you can download it from the homepage I linked to this presentation.
Okay, that's very interesting. Okay, so thank you very much, Professor Vollertsen, for your very interesting talk. I'm sure there will be many sessions during the the panel discussion. So it's time to now invite the next speaker. The next speaker is Dr. Ichi Watanabe, who is the Global Marketing Manager at Frontier Lab, Japan. He received